Have you ever wondered how doctors make incredibly precise decisions about breast cancer treatment, tailoring it just for you? It often comes down to understanding specific details about the cancer itself. One crucial detail is called HER2 status, and it is determined by carefully testing a sample of your breast cancer tissue. This testing helps doctors understand if your cancer cells have too much of a particular protein. The HER2 protein, which stands for Human Epidermal Growth Factor Receptor 2, plays a role in cell growth. When there are too many HER2 proteins on the surface of breast cancer cells, it can make the cancer grow and spread more aggressively. This is why testing for HER2 is so important. The most common way to test is by examining a biopsy or surgical tissue sample from the tumor. Laboratory specialists use special techniques to identify the presence and amount of the HER2 protein. There are two primary methods used for HER2 testing. The first is immunohistochemistry, or IHC. This test uses antibodies to detect the HER2 protein on the surface of the cancer cells and stains them. The results are scored from 0 to 3+. A score of 0 or 1+ usually indicates HER2-negative status. A score of 3+ means HER2-positive. If the IHC score is 2+, it is considered equivocal, meaning uncertain, and requires a second type of test for confirmation. The second main test is called fluorescent in situ hybridization, or FISH. This technique looks directly at the HER2 genes within the cancer cells, checking if there are too many copies. FISH is often used to confirm equivocal IHC results, or it can be used as the initial test. Understanding your HER2 status is incredibly practical for your treatment plan. If your cancer is HER2 positive, it means specific targeted therapies, sometimes called biologic therapies, can be very effective in blocking the HER2 protein and slowing cancer growth. These treatments are specifically designed to attack HER2 positive cells, offering a personalized approach that would not be an option for HER2 negative cancers. It helps your oncology team select the best course of action, which might include specific chemotherapy combinations or other medications. A key insider detail is that sometimes HER2 status can change if the cancer recurs or spreads to a new location. Therefore, doctors may recommend retesting the HER2 status of new tumor tissue if your cancer returns or progresses. This ensures your treatment always aligns with the most current information about your cancer, reflecting its evolving characteristics. It is a dynamic approach to care. Ultimately, determining HER2 status provides invaluable information, enabling your medical team to choose the most precise and effective treatment strategies for your unique breast cancer. This leads to better outcomes and a more personalized journey toward healing, ensuring you receive therapies specifically designed to combat your cancer's distinct profile.